Hello everyone, a very good evening to all of you. Please confirm if you can hear me, if you can see the screen in the chat box. So, since the exam is near, it's just a 1 month ahead. So, how is your preparation going on, everyone? I'm expecting that your preparation will be at highest peak, right? Yes? Am I audible to all of you? Can I get the confirmation from everyone? So when we speak of CSIR NET exam, in that case, we have 3 sections, part A, part B, and part C. So when we consider the scoreable section, that is always part C. But at the same time, even though it is scoreable, at the same time, it is highly risky as well. Risky in the sense you will face negative mark as well. So you have to be very careful while attending the part C questions. So as I told you, this is scoreable. So you should never do any kind of guesswork. Always try to analyze, apply the concept, and then get into the answer. Never try to guess it because as I said you have negative marking over there. So that will reduce the overall targeted score. So in today's session, we are going to take up Unit 2. And under Unit 2, the one of the most important topic that is cell organelles and protein targeting. So every time you will come across questions like 4 marks specifically from this topic of Unit 2. So to start with, here is the first question in front of you. Given below are a few statements about nuclear transport. RAN-GTP levels are higher in the nucleus than the cytoplasm. Nuclear import receptors can shuttle between the nucleus and cytoplasm. NTF2 transports RAN-GDP into the cytoplasm. Export of mRNA is not directly dependent on RAN. tRNA and microRNA export is mediated by exportins. Now you have to identify the correct combination of statements among the given options. Find out. So basically, from this protein targeting part, nucleus is considered to be one of the important cell organelle, whether you consider it in the form of a 2 marks or you consider it in the form of a 4 marks. They are very important. In fact, one of the important organelle. Yes. Can I get answers for this question? So compared to other units, when you speak of Unit 2, nowadays they are asking experimental based questions. Of course, earlier we used to get very much direct questions. Now also we are getting, but in spite of that, we are also getting some kind of experimental questions as well. But that is also not an issue unless and until you are confident with the corresponding concepts. Yes, good evening, Amit. So can I get answer from your side? What will be the answer for this question? Yes. Is it that much tough? This part is that much tough? I don't think so. If you have the basic idea about the nuclear transport mechanism, probably this will be the easiest question you have ever come across. Can I know the answer? Yes, participants, can I know the answer? So the correct answer over here should be, it is your statement A, statement B, statement D, and statement E. Now, why the statement C is incorrect Any idea regarding this? Very simple, just a small mistake in this sentence. What is that small mistake in the sentence? Instead of cytoplasm, if it had mentioned nucleus, it would have been the correct answer. Right? So, C is the incorrect statement among the following. C. NTF2 is a kind of transport factor that is responsible for the import of RAN in the nucleus, not in the cytoplasm. Each importin or exportin mediated transport cycle will be removing one GTP molecule, that is with RAN, from the nucleus and releases it in the GDP bound form, specifically in the cytoplasm. So, any idea why in the cytoplasm this RAN GTP undergoes hydrolysis? Do we have any specific reason why this RAN GTP, upon entering into the cytoplasm, undergoes hydrolysis? Any specific enzyme you are able to recollect? Because we have got two different proteins or enzymes present. So we have got RAN-GEF that is located in the nucleus that helps in the exchange, that is GDP-GTP exchange. On the other hand, in the cytoplasm, we have RAN-GAP, that is a GTPase activating protein. So that helps in the hydrolysis of GTP, and that's the reason you'll find RAN-GDP in the cytoplasm. Whereas the active version of RAN protein, that is RAN-GTP, is present in the nucleus because of this guanine exchange factor, which is tethered to the nucleus, which is tethered to the chromatin present in the nucleus. Okay? And these are the important proteins or karyophorines. Karyophorines are nothing but the nuclear transporters that are responsible for the import and export of proteins either inside the nucleus or maybe outside the nucleus, means taking it outside. So different proteins are involved, like we have importin, which the proteins with basic amino acid, NLS, any protein that is having NLS as their sorting signal will be taken care by importin protein. Then you might have heard about the splicing machinery, right? SNRNPs. Then you have got importin 7. So different types of import proteins comes under this karyophorine receptor. Similarly, we have got exportins. So as in the question, it was mentioned about exportin T, exportin 5, which is responsible for the transportation of tRNA as well as microRNA out of the nucleus. Okay. Now, looking into this import mechanism, how exactly the proteins are getting imported into the nucleus. So, if you see, this is our NLS containing protein. So, corresponding to the protein, you should have a receptor present. And here we have importin alpha and beta serving as the receptor. Any guesses what does this structure indicates? Any guesses? What does this structure indicates? Anyone? So basically it's an import of protein takes place between the cytoplasm and the nucleoplasm. So who is the mediator over here, or through which how the proteins will be transported? Yes. So here, this is nothing but your NPC, that is nuclear pore complex. So through this pore, your corresponding protein will be released into the nucleus. So initially, the receptor and the protein will be tethered to the cytoplasmic filaments present in the NPC, and this cytoplasmic filament will pull the entire content and releases into the nucleoplasm. Within the nucleoplasm, we have got, so now your cargo has successfully delivered into the nucleus. So the next step will be recycling the receptors back to the cytoplasm. So we have RAN GTP that will take up the important beta and releases it into the cytoplasm. Similarly, we have got exportin that will be taking up the next receptor, that is importin alpha, and releases it into the cytoplasm. So this is how the import mechanism happens within the nucleus. Now the next question to all of you. The following intracellular event occurs in a cell that is subjected to conditions of starvation. Now, which of the following statement correctly represents the event shown above? That means this image. And which one is better relating over here? Find out. Akash and Ashish, this is the session for question discussion related to CSIR NET exam. So the questions which you have put, we will look on to that maybe in some other webinar or some other session. So as of now, the CSIR based session is going on, so you can ask any questions related to that. Yes, any answers from your side? Good evening, Vrinda. Okay, I got your answer. What about others? Anyone else would like to give the answer over here? Like nucleus, even lysosome based questions are also being asked in the exam. Of course, compared to the nucleus, the frequency for this organelle is less. So let's have a look into the answer now. So the correct answer over here is the cell is undergoing autophagy by the formation of autophagolysosome. So that represents the C image. Now, what is this image representing? Any idea? What is this image representing? This is a phagophore. That means which helps in the initiation of autophagosomes, right? And if you consider these three proteins, these are the proteins that helps in the initiation, growth and maturation of autophagosomes. So first your phagophore is created. Next, your autophagosomes are completed. That is, the double membrane structure is completed now, which is engulfing the target organelle or any substance that is not needed by the cell. And now comes your B, that is your lysosomes. So now A and B combines together in this way, and what we are getting? Autophagolysosomes. Now, lysosomes is having the hydrolytic enzymes, so they will be targeting the content for degradation. I hope so you got it. See, autophagy genes like 12 can form complex with autophagy 5. And they are basically involved in the formation of autophagosomes, basically for the elongation purpose. Beclin 1 is also the same thing. They are involved in the initiation stage and they also help in the recruitment of other proteins in autophagy. Specifically, you might have heard about the PI3 kinase, right? So that is being recruited over here. LC32, this is considered to be a marker enzyme for autophagosome. So based on these understanding and based upon the sequence of steps, we can identify 2 as the answer. So autophagy means where we are digesting the material that is of intracellular origin. That means whatever the old organelles are there, it has to be replaced with fresh and more effective ones. For that purpose, you have got the process autophagy running. So in this case, as I told you, first a membrane is donated by the ER. We call it as a phagophore. Now, this membrane will be surrounding the old organelles. Okay. Now, you will find that a whole structure is constructed, which we call it as an autophagosome, which combines with the lysosome, forming the autolysosome. And after that, the degradation happens. Okay, so this is how it goes. The next question. Based upon, so following is the domain organization of three proteins that are targeted to the mitochondria. Now, based on the domain organization in the figure and assuming the left box to be having the mitochondrial sorting signal, predict the most likely subcompartment of the mitochondria in which the protein will be found. So, this is the image given to you, and you have to identify where this protein A goes, where the protein B goes, and where the protein C goes. Now, how you can identify? Based upon their sorting signals or based upon the polypeptide chain. Try to find out. Try to find out the answer for this question. So mitochondria specifically, it is having different compartments, right? It is having outer membrane, inner membrane, intermembrane space, mitochondrial matrix is present. So based upon this, questions do come in the exam. As of now, they have not asked any experimental question based on mitochondria. Yeah, they might be asking, but again, it is a concept based only. You will be able to get into a conclusion for sure. So what should be the answer for this question? You can easily identify this. See, the first image is showing a mature protein and it is having matrix protease cleavage site. So obviously it will be directed towards the matrix, right? If it is not going to the matrix, what is the use of this MTS sequence? So matrix protease cleavage site is there. So we can easily say that option 1 is the correct answer. Now coming to B. B is inner membrane bound protein. How we can say that B is an inner membrane bound protein? Because it is having a hydrophobic region through which it can tether to the membrane. It is having the inner membrane protease site and it is having some exposure to the matrix cleavage site as well. So because of this, we can easily say that it is present in the inner membrane. And next we have the intermembrane space. So intermembrane space, obviously those proteins can interact with the outer membrane import pore. So we can get into a conclusion that A is for matrix, B is for inner membrane, and C is for intermembrane space. Okay. These are the signal sequences, which if you able to recollect, that will be easy for you to score better. So if you can see for matrix, you have an MTS, which is a part of the mature protein. For inner membrane, you have got different types of polypeptide chain. It can have the exposure to matrix and it can have some hydrophobic sequence that will help in tethering to the membrane. Some proteins will have internal sequences which will be recognized by OXA1. Now, OXA1 is basically a protein that is responsible for carrying the proteins to the inner membrane if it has moved to the matrix. And then you have got the pathway C, where they will be having only the internal sequences present, and those sequences will be recognized by the receptors present in the outer mitochondrial membrane. Okay. This is for intermembrane space. You have got first cleavage by matrix protease. That means again it is having exposure to the matrix region. Then second cleavage that releases your corresponding polypeptide into the intermembrane space. And the second pathway, it does not have any MTS, so there is no connectivity to the matrix. It will be directly moving to the intermembrane space by means of general import pore. And for outer membrane, if you see, they will be having just the outer membrane localization sequence through which it will be located in the outer membrane itself. Okay. Moving on to our fourth question. A null mutation is created in a gene which is responsible for specific phosphorylation at 6th carbon position of mannose on acid hydrolases that occurs in cis Golgi compartment. So here are some statements given below to explain the effect of mutation. Lysosomes will be devoid of lysosomal enzymes. Lysosomal enzymes will be secreted out, or lysosomal enzyme will be get localized in the cytoplasm. Now you have to identify the correct statement or combination of statements that will explain the effect of mutation if the acid hydrolases in the mutant do not get degraded. Identify. Try to identify the correct answer. See, unless and until you try on something, how will you understand that you are confident or not confident? See, giving a right answer, wrong answer is a separate thing. But first, you try to give the answer. Who knows what the concept you are holding on is applicable over here, and you'll get a correct answer. See, this is not even a CSIR NET exam where you have to think a lot, and then get into a conclusion. You're not losing any marks over here. This is just a session where you are trying to analyze yourself, what you are strong enough, in which portion you are strong enough, and in which portion you are lacking that confidence. So try to give the answers, everyone. Yes, can I get some answers this time? Yes, Anusha. Okay, what about others? Try on, everyone. Unless and until you try, you will not get into a conclusion. Try on. Yes, can I expect some more answers for this question? Yes, some more answers for this question. They are simply saying about a mutation that has happened in the 6th carbon position of mannose residue. Just tell me once, which is the only cell organelle where carbohydrates will be serving as a sorting signal? Majority of cell organelles you take into consideration, their amino acids are considered to be a sorting signal. But there is only one cell organelle where sugars will be considered as a sorting signal. Which one is that? Absolutely correct, Vrinda. So now connect it over here. Connect your answer to this question. What should be the answer here? They are asking about the effect of mutation. What will be the consequence of this? Null mutation. So the correct answer over here is A and B. So the lysosomes will be devoid of lysosomal enzymes, and lysosomal enzymes will be secreted out. Now, why the lysosomal enzymes will get localized in the cytoplasm is incorrect over here. The reason is that have you heard about co-translational import and a post-translational import mechanism? Have you heard about this co-translational and post-translational import mechanism? Are you aware of that? Anyone? Any one of you? What is the difference between them? Because based on that, you can eliminate the statement C from here. First thing is that they already have a mutation in the mannose residue. So obviously they will not be targeted to the lysosomes. Okay. So what will be the situation then? See, if the mannose group is not phosphorylated at the 6th carbon residue, then lysosomal enzymes will not be targeted to the lysosomes, as this is the sorting signal for its entry inside the lysosome. So lysosomes will be devoid of lysosomal enzymes. Also, since they will be present in the Golgi, these enzymes will be secreted out of the cell. Now, the two terms which I said you, co-translational and post-translational import. See, co-translational import in the sense, as the name indicates, at the time of translation itself, it will be deciding or retained within the organelle, that is, within the endoplasmic reticulum. The synthesis is not complete, but before the completion itself, it has found a cell organelle for that. After that, it will be released completely, and then it will be deciding whether it has to retain in the ER or it has to move to the Golgi. If it moves to the Golgi, obviously it will be secreted out, either in the form of secretory vesicles, lysosomes, or maybe membrane. On the other hand, if you take into consideration post-translational import mechanism, that means after translation only the protein will decide what to do. So the translation takes place in the cytoplasm, and after the complete synthesis, it will decide whether it has to remain in the cytoplasm or it has to be imported into any of these 4 cell organelles. Okay, so that is the difference between co-translational and post-translational import. And that's the reason I told you that the statement C is incorrect over here. Okay, I hope so you got it. Moving on to our next question now. The tetrapeptide KEDL is known as a retrieval signal of several newly synthesized proteins. This process is mediated through specific receptor, that is KEDL interaction. Any single amino acid change in this tetrapeptide is not allowed in terms of its binding with its receptor and its subsequent retention in specific organelle, whereas the secretory proteins are devoid of such tetrapeptide. So from this observation, indicate the localization of the receptor of this tetrapeptide. So where exactly the receptor should be present? Plasma membrane, Golgi, ER, or mitochondria? Okay, that's good to hear from you, Anusha. You understood that point. I hope so, Vrinda, you also got it? Okay, fine. Yeah, first tell me, KEDL is the signaling sequence for which cell organelle? KEDL is the signaling sequence for which cell organelle? We have so many cell organelles, and different cell organelles are having different sorting signals. Absolutely correct, Vrinda. So it is considered to be an ER retrieval sequence. Yes, Anusha. So what should be the answer over here then? So the KEDL receptor will be present where here? According to the question. So now that you have already mentioned that it is present in the ER and it serves as the retrieval sequence of ER. So what do you think? Where does this receptor will be located? KEDL receptor. In which cell organelle? Okay, Vrinda. So Anusha, ER is your answer? Anyone else would like to give the answer? For this question? After the nucleus, the next cell organelle, which is considered to be very important from examination point of view, will be the endoplasmic reticulum. You'll get a lot of questions based on this. Okay, Anusha. So I got the answer from Vrinda and Anusha. Very good. Very good efforts. Can I expect some more answers? Can I expect some more answers from other participants as well? So the correct answer over here is Golgi, and you both have said absolutely correct. Is there any specific reason why did you mention as Golgi, although you know that Kedal sequence is specific for ER? Why did you mention this as a Golgi? Any specific reason? Yes. The reason is that the Kedal receptor binds more tightly to its ligand at the low pH. And it is thought that, yeah, retrograde transport, that is fine. But the major part is that the ligand and the receptor will be tightly bound at low pH, which you will find in the Golgi compartment. And it is thought that the receptor is able to bind the Kedal peptides in the cis Golgi and release these peptides in the ER because the pH of the Golgi is slightly lower than that of the ER. So you will find that in the Golgi compartment, it will be below 7, like 6.4, 6.5, like that. But if you see ER, they will be having this near neutrality pH, that means around 7 or sometimes more than that. So that's why the Kedal receptors resides in the Golgi compartment. Okay. So a second sorting signal that target proteins to COP1 vesicles and also enables the recycling process could be diarginine sequence. So it is not only the Kedal is the only sequence that serves as a retrieval sequence. In certain cases, you will also find diarginine sequence can also do the same function. And this KKXX is something you call it as a membrane bound signal. That means those membrane proteins. So this must be located somewhere in the C terminus of a protein. The diarginine sorting signal can reside in any part of the membrane protein that is present on the cytoplasmic place. So when you consider ER, it is not necessary that the sorting signal should be present in the N-terminus. It can be present either in the N-terminus or it will be present in the C-terminus as well. Now, based on that, just try if you can solve this question or not. So you have some cell organelles given under column A, and then you have some specifications related to that cell organelle in column B. Try to identify the correct combination from here. What should be the correct answer or correct combination over here? Okay, Vrinda has given the answer. What about others? Anyone else? Anusha and others? Would you like to give any answer over here? So you have got lysosomes, you have got cis Golgi, trans Golgi, COP2 vesicles, and endocytic vesicles. One thing, if you are sure of COP1, COP2, and endocytic vesicles, that means what are they and what is their function, you'll be able to get into a conclusion over here. You'll easily fetch a 4 marks out of this. Yes, what should be the answer over here? In your Lordish book, you have got a table that defines these 3 types of vesicles. COP1, COP2, and endocytic vesicles. Anyone else would like to give the answer for this question? So basically, when it comes to Unit 2, you can expect three kinds of questions. One is this kind of match the following kind of questions. You can also expect in the form of statements. Okay, you can also expect statement based questions as well. Then experimental, as I told you in the recent past, you are getting some questions over here. Okay, Vrinda, you have changed your answer to 1 now. Okay. Any other answer from anyone else? Okay, let's check out the answer now. The correct answer over here is absolutely correct, Vrinda. It is 1. First thing is that when you get this kind of question, check out the odd man out if you can. The first odd man out is we are pretty sure that lysosomes is the only cell organelle which will be having sugar molecules playing an important role. So we have got mannose 6-phosphate receptor relevant to lysosomes. So in that case, what happens? You can easily eliminate options 2 and option 3. Now what you are having? Option 1 and option 4. Now you have to identify which is the next important concept. Have you heard about this anterograde and retrograde transport mechanism? Have you heard about it? Have you all heard about anterograde retrograde transport mechanism? Yes. So basically anterograde is something that moves in the forward direction, that is ER to cis Golgi compartment. On the other hand, as mentioned by Anusha earlier, retrograde means they are moving in the reverse direction. That means from Golgi to ER. Right. Now you have to connect this with the type of vesicle that carries the process. So what you are having over here? COP2 vesicles. That helps in the movement of proteins in the forward direction. So now you got COP2 vesicle, anterograde movement, D21. So here also you are having D21. Then next is the cis Golgi network. Cis Golgi network. So you are aware that COP2 vesicles originates from the ER. That's why it moves in the forward direction. So the cis Golgi network means they will be releasing COP1 vesicles and they will be carrying out reverse direction. That means Golgi to ER. So now you got cis Golgi to 3 also. Then you have got the trans-Golgi network. Trans-Golgi is something which I already mentioned that they will be secreted out. So protein aggregate for secretion. Then you have got the endocytic vesicle which is related to the clathrin-coated vesicles. Okay. So try to analyze each and every cell organelles with respect to their corresponding functions, whatever is given in next column. So this is a image that makes you understand the retrieval process. So you can see that this is your endoplasmic reticulum. So obviously you have got ER resident proteins present. That is indicated in violet color. Now from here, the COP2 vesicles will be released. By mistakenly, what does it do? It carries the Kedal sequence containing proteins and releases into the cis Golgi network. No problem, because we have got Kedal receptors present in the cis Golgi network. And from here, the COP1 vesicles will be pinched out, which carries the Kedal sequence containing protein and retrieves it or releases it back to the ER. So this is how this retrieval process works. And the table which I had mentioned was this table. COP2 vesicles, ER to cis Golgi. The accessory protein or the coat proteins are this. Obviously, they require some sort of energy for the transportation process. So the GTPase over here is SAR1. Rest all are ARF. For COF1, you will see cis Golgi to ER. For clathrin and adapter proteins, we can have trans Golgi to endosome, where the clathrin is bound with adapter protein 1, or it could be from plasma membrane to endosome, where the clathrin is bound with adapter 2. That is depending upon the location, the adapter proteins vary. But the associated GTPases remains the same, that is ARF. So this table is important, and based on this vesicles, many a times questions had come in different formats. But the thing is that if you're clear with this concept, you can easily fetch a 4 marks out of this. So just a reminder to all of you is that we are starting with a new batch, that is Lakshya batch, that is starting from 10th of January for all the CSIR NET aspirants and the GATE 2026 exam. And we will be having, like this course will be providing you with unlimited classes, 24/7 chat support system. You'll be having a study portal where you can access all the PPTs and recorded lectures. Even if you have missed it out, no problem. You can always check on to this study portal. Then some smart tools will be there that will help you to revise your topics in a more better way, in a concise way. We will be having regular test series, weekly test, monthly quest. Apart from that, some in-net test series are also there. That is, unit-based test series will be there. And the one of the, what I can say, another advantage of this lecture batch will be you are getting a free molecular biology internship as well. Right? So you all might be aware how much important is molecular biology when it comes to CSIR labs. So you will be trained on that as well, and that too free. So if you want to know more details about it, contact us with this toll-free number. We'll be getting more information about the same. Moving on to our next question. The following statements we have made regarding the role of protein modifications. Attachment of acetyl group to the N-terminus of the protein makes it more resistant to degradation. Attachment of hydroxyl group to proline residue stabilizes the fibers of newly synthesized collagen. Glycosylation, that is addition of sugars, will make the protein more hydrophilic, enabling protein to protein interaction. Or addition of sugars will make the protein more hydrophobic, enabling protein folding. Now you have to identify the correct combination of statements over here. Find out. So basically, protein modifications happen in which cell organelle? This glycosylation and all will happen in which cell organelle? Specifically, the glycosylation and all. You are aware, right? We have two types of glycosylation, N-linked and O-linked. So what are the cell organelles where this process will be carried out? Yes, you are correct, Vrinda. So just try to get an answer for this question. Try to get some one answer for this question. You have to identify which protein modification mentioned over here is correct. No idea? Do you know the significance of acetyl group, significance of hydroxyl group on collagen protein? You know about the structure of collagen protein? See, the correct answer over here is first three statements are correct. So whether it is your acetyl group, whether it is the hydroxyl group or the glycosylation process, all the three are correct. So glycosylation will be inducing hydrophilic interaction. So it makes the protein more hydrophobic. That is incorrect over here. See, acetylation is a kind of post-translational modification where acetyl group will be added to the N-terminus of a protein. Specifically in which amino acid? In the lysine residues. So what does this lysine residues will do, or what is the significance of lysine residues getting acetylated? They will neutralize the positive charge present on the N-terminus, which makes it less susceptible to cleavage by any sort of proteases. And this acetylation can also lead to the formation of protein to protein interaction that will stabilize the protein and protect it from any sort of degradation. Then, regarding collagen, hydroxylation is a very important process. Specifically, this is a triple helical structure, right? The collagen, you might have heard about it. So, how this triple helical structure is maintained? Because of the repeats glycine XY. And what does this XY indicates? X could be, if it is proline, it will be hydroxyproline. If it is lysine, it will be hydroxylysine. So hydroxylation is a major process contributing towards the collagen structure. Next, glycosylation makes the protein more hydrophilic because sugars are polar. They will have hydrophilic properties. Hydrophilic means that will be attracted towards water. So when glycans will be attached to a protein, they can create a hydrated shell around the protein, thus making it more soluble in the aqueous environment, both inside and outside the cell. Absolutely correct, Vrinda. Yeah, you are speaking regarding the collagen protein, right? Ascorbic acid, yes. So this increased solubility can enhance the protein's interaction with other molecules, including other proteins. Okay. Okay, then. Moving on to our next question. To delineate the steps in ER transport, now this is an experimental question. A PhD student homogenized pancreatic SNR cells to isolate microsomes, which retain most of the biochemical properties of the ER. For this experiment, the student has planned a number of controls as mentioned below. Treat one set of microsomes first with detergent, then with proteases, or with protease only, or with nucleases, or with detergent only. Now, they have provided you with some combination of controls, like it could be A, B, and D, B, C, and D, A and C, B and D. So you have to identify the correct combination of this. Check out the statements here. Then you should have answered it, Anusha. See, that's what I said you. Whether you are saying right or wrong, no problem. Just give the answer. See, if you are right, you'll get the confidence ahead that yes, I know something and I can make myself better. And if incorrect you are giving, then you are rectified, right? So you will not make the mistake again. So there is no problem. Always try to give the answer, specifically in the sessions and all. Of course, when you go for final CSIR NET exam, you have to think and then get into an answer. But when you are trying to solve the questions by your own or sessions like this, try to interact and try to get the answer. Because we are here to help you out. If you are not able to understand a particular thing, we are here to help you out. And that's why we have these kind of sessions. Yeah. Can I get answer for this question? Yes. The what is the combination of controls? No idea. See, this is a very simple question. How? See, the correct answer over here is A, B, and D. So, why we are eliminating the nucleuses out of this? What is the function of nucleus? Can anyone tell me? What is the function of nucleases? And what is the function of proteases? A very simple question to all of you. Because based on this only you got an answer. What is the role of protease enzyme? To target the proteins for degradation. Yes. And if I say nucleus, they will be targeting DNA, obviously. So in this case, here we are speaking about proteins. So is there a necessity for nucleus digestion? No. So you can easily eliminate this one. And detergents and proteases are obviously required for any kind of proteins to be extracted. Right? So that's why you have got A, B, and D as the answer. See, microsomes are a heterogeneous set of vesicles which are obtained from the ER when the cells are disrupted. These vesicles are isolated by differential centrifugation and composed of three features, that is rough vesicles, smooth vesicles, and ribosomes. Microsomes are the most widely used preparation of ER, and it is used for co-translational processing of membrane proteins. When I say detergents, obviously they will be lysing the cells. So the soluble proteins will be released. Solubilize the membrane proteins and lipids. Proteases, these are nothing but the enzymes that will be breaking the peptide bonds of proteins. So when I say control, all the three can be used individually as well as in combination. But nucleuses, as I said you, they are something that is targeting the DNA and they don't have any role over here. So we can easily omit the nucleus part. I hope so you got it. Yes. So in that case, proceed with the next experimental question now. Isolated mitochondria were either treated with protease or first briefly incubated in hypotonic solution prior to the treatment with proteases. The reaction was stopped and samples were probed for the presence of MTG2 protein, porin present at the outer membrane, cytochrome C present in the intermembrane space, KDH present in the matrix using Western blot analysis. Now, based upon the gels given over here, you have to identify the location of MTG2. Identify. So here you have to analyze the blotting. What is happening in the intact mitochondria and what is happening in the mitochondria that is placed in the hypotonic solution. What is the difference among them? And what about this MTG2? You have to just analyze understand the image properly. You can easily get into a conclusion. Other proteins location is given over here. See, porin present in the outer membrane, cytochrome C present in the intermembrane space, KDH present in the matrix. Right. Now you have to identify what is the location for MTG2. So based upon this immunoblot analysis, how can you identify? Okay, Vrinda, I got an answer from your side. What about others? Any other answers can I expect? Okay. So the correct answer, you are absolutely correct, Vrinda. It is matrix. So how did you identify that this is a matrix-based protein? The thing is that here it is mentioned KDH, right? KDH is present where? In the matrix. So if you see the KDH which is present in the intact mitochondria as well as the mitochondria placed in the hypertonic solution, almost the bands are visible. So the protease has not attacked over here. But if you see porins, it is completely vanished. Cytochrome C, it is vanished. But MTG2 again remains intact. Right. So this indicates that they must be occupied somewhere within the matrix. See, MTG2 is present in the matrix because according to the blot analysis, the KDH protein shows the same profile when treated with protease alone or when treated with the hypertonic solution. So in the intact mitochondria, this particular protein, even after the protease treatment, although cytochrome C and porin level decreases, but it remains intact. KDH also remains unaffected. And the same result you saw in the hypertonic solution also. So based on this, we can say that yes, option 3 is the correct answer. So this is the last question of today's session. A Kedal sequence is added to the C-terminus of a secreted glycoprotein X having no site for N-linked glycosylation and expressed from an inducible promoter. Following 10 to 20 and 60 minutes of induction, ER is purified and probed for newly synthesized glycoprotein X Kedal. Now the immunoblot is given over here. Now you have to identify the reason why in the 20 and 60 minutes the molecular weight of you can see width of the band is higher. What is the reason behind it? The signal sequence is not removed from some of the glycoprotein X Kedal molecule, or it has been modified in the ER after protein synthesis is completed, or the corresponding protein is modified in the Golgi prior to the ER retrieval. The quality control mechanism in the ER recognizes a pool of protein as aberrant or targeted for degradation. Identify the reason over here. See, based on these kind of blot analysis based questions, you can always expect in the exam. And I don't think so this is that much difficult. As I can see, Vrinda is easily giving the answers. And most of her answers are also correct. Just you need some time to think and analyze. That's it. Okay. Anyone else would like to give the answer? So the correct answer is 3. Now, why the answer is 3 given? Why do you think that option statement 3 is correct? Because the molecular weight increases, that indicates what? That indicates that obviously some kind of addition takes place. Some kind of extra sugars might have taken place. Sugar addition. That's the reason they are mentioning option 3 as the answer. See, the Kedal sequence is considered to be a retrieval sequence that directs the protein back to the ER from the Golgi. So when a protein with a Kedal sequence by mistakenly transported to the Golgi, it has to be modified before being sent back to the ER. So this appearance of higher molecular weight bands over the time indicates that the corresponding protein has underwent some kind of modifications over there. And you know very well that modifications can happen in the Golgi, right? So after the modification, the protein will be retrieved back to the ER. Why? Because it is having the Kedal sequence present. So you can expect these kind of questions every time in the exam. And as the CSIR exam is coming, and every time whenever you go for the exam, the questions are getting more tougher and tougher. So try to practice more these kind of questions so that you will learn how to approach a question. So with this, I'm winding up the session and just wanted to mention that you'll be provided with some questions to work out. Try working out on yourself and then check the answer. And tomorrow also you will be having the session based upon CSIR NET syllabus, do attend the session. Okay then. Thank you so much, everyone.